All right, everybody, let's run through the review for chapter 2. So chapter 2 covered chemistry and basic rundown of chemistry and the concepts therein. We talked about the different kinds of bonds, covalent bonds where we're sharing electrons, ionic bonds that involves, one atom transferring an electron to another, one or more electrons to another atom. Like our example that we gave was sodium chloride. So no, forget every time. I apologize, you guys. I forget. Okay, so we have sodium chloride, and then the sodium gives an electron to the chlorine. So that would leave the sodium with a little positive charge because we lost a negative charge. And then it leaves the chlorine with a negative charge because it gained a negative electron, right? So that's how that would happen. So these guys are associated with one another via that ionic bond. You pop these into a solvent like water, and they can ionize, essentially. So ionization, the sodium, the positive is going to associate with the oxygen of an H2O molecule because that tends to have a negative charge to it. The hydrogens tend to have positive, and that's what the chlorine would be associated with because of its negative charge. Okay. And that's why these guys are happy in solution, because of that polarity. Polarity refers to that charge that we've developed there. So polarity can, if something has a charge like that and we have swayed to a positive or we have swayed to a negative, usually due to hogging of electrons one way or another, in that covalent bond, when we hog an electron from a hydrogen and it gets its little positive charge like we do here, and then we get our negative charge on our oxygen, that's due to the electronegativity of the oxygen. So it wants to pull the electrons to it. So that's created that polarity. That's polarity right there. You have that charge going on. Now, if you have something like a phospholipid, this long chain here is just hydrocarbons, and that literally just means hydrogens and carbons. And that's it. It's just a whole bunch of that. This tends to be non-polar. So this portion would not interact well with water. So we would say it's water-fearing. That's the way that we can define the word, but the official science word would be hydrophobic, right? So if we look at something like sodium, or we look at chloride, the chloride ion, or if we look at the head associated with this, which is a phosphate, which has oxygens, and I don't know how many bonds are associated with it, so don't worry about that. We just know it has oxygens, right? And we know that the oxygens, because we already learned how it's electronegative thanks to the water, we know that these guys are going to have their negative aspects to them. So they're negative. They're pretty negative. So this guy has a charge. So if we're just talking about the head of this here, this is polar. And just the head, this would be hydrophilic, like Philadelphia, city of brotherly love, where I went to school. So that sets you up for that concept of the what hydrophobic and hydrophilic are. Phobic, phobia, fear of water, right? I was thinking Old Yeller. Probably should not even talk about that because I just feel like it's super depressing, but whatever. He has hydrophobia. That's what they say in the movie. So because of rabies, just FYI, in case you haven't seen it. So we have our polar and our non-polar molecules here, hydrophobic and hydrophilic. However, I've already told you this is going on in one molecule. So is this molecule our phospholipid? Is this molecule hydrophobic, or is the molecule hydrophilic? Is the molecule polar or non-polar? Well, there's a word for the molecule, so you don't even have to worry about it. When they have both going on, which is what's happening here, we can agree on that. because we have the polar and the nonpolar. We have the hydrophobic and the hydrophilic. We have both of those things happening. The word for this is amphipathic. In general, when you hear amphi in this course, this won't be the last time that you'll hear it, by the way. In general, when you hear amphi, in general, both, means both. And if you ever have a hard time remembering that, think of amphibians, like frogs, that can do both the water environment as well as the land environment. So that's exactly what we're talking about here, right? Both are happening. Okay, so we have talked about our, we talked about our sharing. We talked about ionic bonds. A hydrogen bond, we talked a little about this. So the hydrogen bonds basically are occurring when we have creation of these polar aspects to these molecules. So the oxygen being electronegative and hogging the electron from the hydrogen. The hydrogen gets its positive charge, the oxygen gets its negative charge, but it's still just a covalent bond. They're still bound together, sharing electrons, just the oxygen hogs it more. So whenever that positive hydrogen interacts with something else that also happens to be negative on another molecule, that is a hydrogen bond. They are weak bonds, and they happen due to the hogging of electrons, like we see happening in covalent bonds like this one, polar covalent bonds, due to the electronegativity of a molecule. Cool. So we've talked about that. We talked about polarity. We just talked about that, amphipathic, hydrophobic, hydrophilic. Organic molecules are any molecules that have a carbon-hydrogen backbone. If it's only got carbon just kind of by itself, then that's not considered to be organic. That would be inorganic. Anything that does not have a carbon-hydrogen backbone is going to be inorganic. The rest is organic. pH, the concept of pH. Remember that pH is a logarithmic scale. So we are talking about concentrations of hydrogen ions. And again, that's going to be the H+ ions in a solution. Hydrogen ions. So a measure of the concentrations of these guys. In terms of 10 to the negative something. So 10 to the negative 2 versus, 10 to the negative 10. Now, if you're looking at these just as numbers, 10 to the negative 2 as a number and 10 to the negative 10 as a number, which one is the smaller number? Which has less? If this was money, which would you rather have, right? So 10 to the negative 2, 10 to the negative 2 is greater than 10 to the negative 10. That means that the hydrogen concentration is greater. So this is a measure of hydrogen concentration, by the way. And so logarithmic scale teaches us that we're going off of this number here. So the concentration might be 10 to the negative 2. The pH would be 2. This pH is 10. So that's how that works. So the lower numbers are more acidic. They have a higher hydrogen ion concentration. The higher numbers are more basic or more alkaline. They have a lower hydrogen ion concentration and, importantly, a higher hydroxyl concentration, because all of this is really coming back to H2O and this kind of setup. This is a negative on the oxygen. So we're just talking about measuring concentrations of this versus this when we're talking about pH. And pH, the number itself, is really directly looking at this. Okay. So that is pH and how that works in a nutshell. I say that all the time, and I said it again. Okay, the macromolecules that we talked about, the carbohydrates, this is dealing with sugars. If we talk about a saccharide, a monosaccharide is the monomer or the polymer being a carbohydrate, essentially a polysaccharide. So something like starch, if we're really going to get into examples of it, or cellulose, which was the example that the chapter gave us. So the sugar-based molecules. Then we have the lipids. Those are the fats, like the triglycerides, storing energy for us, as well as the like sterols that are giving support and structure, and the waxes giving protection of cells to things like tuberculosis. Then we have the nucleic acids. This is our genetic information. So this is going to be DNA and RNA. Remember that DNA had, they both were made up of nucleotides, first of all. So that is a 5-carbon sugar, a pentose of some kind, a phosphate group, and then a nitrogenous base. So that's a base that has nitrogen in it. And that's going to be either adenine, guanine, cytosine, or I lost A. I didn't say T. Thymine. Okay. I was like, well, I should have wrote them down as I went. So I couldn't remember which one I said already. Thymine is the T. So that's for DNA. If you want for RNA, it's going to be adenine, guanine, cytosine, and uracil instead of thymine to replace that with the U. So that's the difference for the nitrogenous bases. They also have different sugars, RNA and DNA. DNA has deoxyribose and RNA has ribose. So it's literally deoxyribose has a OH group that gets replaced with just an H. That's been deoxy. Surprise. So that's the differences there. The DNA is going to be like your genetic information. Like the example that I gave was that's the information stored in the actual Smithsonian. You wouldn't take that out of the archives of the Smithsonian. So you make a copy of what you need and then take that out of the Smithsonian. So you can make, whatever thing from that blueprint, right? So that's exactly what's happening. So DNA is a master copy. RNA is our working copy. RNA also happens to make up the structure of the tools that we need to read the RNA and turn it into a protein. So that's interesting stuff as well. So that's the idea of those guys. Then we talked about proteins. Proteins are made up of repeating units of nucleic acid. Sorry. Oh my God, I'm making you real confused here. Proteins are made up of repeating units of amino acids. So remember, the amino group is the NH2 group. And then they also have the acid part, which is the carboxylic acid. And those can bind across with one another, next to each other, next to each other, next to each other in bonds called peptide bonds. And that's what makes up a polypeptide. And so the primary structure of a protein is just going to be that amino acid sequence that results. Secondary structure is going to include our alpha helices and our beta sheets. Those are going to start forming due to the polarity or non-polarity of the R groups, which are what are different and make them different amino acids. And then we have the tertiary, which is like even more associating with, the larger aspects of the folding of the protein due to hydrogen interactions, hydrogen bonding, as well as disulfide bond formation. And then we have the quaternary structure, which is involving, multi-polypeptides interacting with one another to make a functional protein. And that is what protein structure is. Then we talked a little bit about cells and what cells are and how they are the fundamental units of life. And that is basically that was it for chapter 2. And that was the chemistry chapter. And I don't know, I'll see you guys next time.